Hello everyone. Today we have a very special guest with us, Professor Mohit Kumar Jolly, who is an associate professor at the Department of Bioengineering in Indian Institute of Science. He did his BTech and MTech from IIT Kanpur, and he did his PhD in bioengineering from the Rice University in US. Followed by this, he also did postdoc and then joined Indian Institute of Science, where he is currently leading the Cancer Systems Biology Lab. His focus is multifaceted, and in today's talk, we'll be focusing on the cancer drug resistance aspect that his lab is currently working on. So this topic is one of the crucial or the niche topics that leads to most of the fatalities in addition to metastasis. So we'll be asking Professor Mohit Kumar Jolly some insights from the current work that he is doing and also what is the current understanding in the field. So thank you very much, Professor, for joining, and we look forward to hearing your insights in this talk.
Thank you so much, Anjani, for the kind invitation and the introduction, and really looking forward to interacting further and discussing many of these open questions which have a great societal value as well.
So before starting, I would like to understand what is the cancer drug resistance paradigm and why the cancers evolve drug resistance.
Sure. So given different therapies that are being used in the clinic right now, not every therapy works for every patient, at least as effectively, right? So the objective of these therapies, of course, is to make a given patient cancer-free, not just at that point of time, but also for as prolonged periods of time as possible. So different therapies have and different patient combinations are good at reducing the initial tumor mass or initial tumor volume. But in many cases, what happens, unfortunately, is that the tumor mass again begins to increase after a few weeks, months, or years, depending on which scenario we are talking about, and which is what is called as tumor relapse, which largely happens because the same therapy that was initially able to control tumor mass is now no longer effective, which is what broadly is called as drug resistance. Now, drug resistance as a dynamic process, this can happen in at least two ways, and also a combination of them. One is that there were some cancer cells initially which were not responding to a given therapy or a given drug because their perhaps molecular footprints were different. Molecular makeup was different, and this drug was not able to impact the function as efficiently. Or the second approach is that every cell was perhaps sensitive to the drug to begin with, but some cells had this ability to change their behavior in such a manner such that now the drug no longer impacts them. And I think most of the studies that have come out in different cancers and different drugs combinations show that it's often a mix of them, that there are some sort of pre-existing resistant cells, but at the same time, cancer as a group, some cells in that group have this ability to switch to a different behavior, a phenomena that is called as phenotypic plasticity, where they change their phenotype or their functional behavior during the application of the drug itself. So it's a mix of both of them. Now, these changes can happen again at least two different levels. One is genetic and the other is non-genetic. Genetic, as we all know, refers to changes in the DNA sequence itself, which can be passed on to multiple progenies coming from that individual cell. It's an inheritable process and it's an irreversible process. But non-genetic means that there is no change in the sequence of the DNA itself, but there can be other changes, such as the presence of different marks on the DNA sequence itself, which are reversible and which may or may not be inheritable. So these are usually much faster changes, much more reversible changes. And again, in multiple studies we have seen as a community, we observe that it's a mix of genetic and non-genetic or phenotypic adaptation processes that cancer cells are able to take in order to evolve to that stage where the same drug is no longer working as effectively as it worked initially.
Very interesting. So here you delineated basically the two different mechanisms through which the cancers can acquire drug resistance. One is the genetic, other is the non-genetic. So as you spoke about, different mutations may be present in the cancer cells, which might be conferring them with drug resistance. Uh, to me, what is more interesting is the non-genetic mechanism, because you said it can be also reversible. So the cancer can use it at its will. So, for example, when it encounters certain drugs, it can use that mechanism to shield it against the drug. But at a later point, when it doesn't require, it can switch to other normal or some other state. So, could you also delineate what are the different cell types that are seen in the common cancer microenvironment, and how the phenotypic switching happens? What could be the underlying mechanisms that could be leading to this phenotypic switching?
Certainly. So phenotypic switching or phenotypic plasticity can happen in multiple manifestations. I mean, cells can change their metabolism, for instance. So the metabolic state can be different, which can be influenced again by the environment around them in terms of how much oxygen is present, in terms of how stiff the matrix around the cells is, and so on. It can also happen that they can change their, say, migration or invasion behavior. They can change again reversibly. They can stop being in active cell cycle stages and rather become quiescent or dormant for certain periods of time. So these are all different manifestations, and these are not independent, not completely independent axes. These behaviors are connected. So the changes in migration or invasion usually accompany changes in metabolism in a specific direction and vice versa. So what we have been trying to investigate is what are the different exit routes that a cancer cell has given a particular drug. We have taken multiple examples of this. In breast cancer, for instance, we have been looking at how resistance to tamoxifen, which is the first targeted therapy that came into the markets in 1980s and is given to what is the most common subtype of breast cancer called estrogen receptor positive breast cancer. So again, it works very well initially in terms of reducing the tumor mass, but then at least based on some clinical observations, one third of the patients come back with metastasis after usually about a decade or so, and then tamoxifen, the patients no longer respond to tamoxifen. So here you see metastasis and drug resistance properties happening, co-occurring together, again after almost a decade or so.
Yeah, so that's very insightful that you give the example of tamoxifen resistance. I'm assuming that there would be other drugs which the cancer would have become resistant in different cancer types. So, sticking to this breast cancer and tamoxifen, you in your previous papers have also talked about hybrid states and epithelial mesenchymal states, this switching. So, could you please delineate what could be the impact of hybrid states on becoming drug resistant in future, and how are they different from the normal known epithelial and mesenchymal phenotypes? What makes them different from the standard cell types, if we may say?
Sure. So initially, this process of epithelial to mesenchymal transition, or these two broad speaking cell types, was considered, or I would say assumed, as a binary process, that cells are either epithelial, which means that they are stuck in their uh, environment with tight cell-cell adhesion bonds and they do not have the property to move, or they are mesenchymal, which means that their cell-cell adhesion bonds are very weak and they can migrate and invade at their own will. So some of the initial work we had done asking this question, that is it really a binary process, and the answer we got based on mathematical modeling of the underlying interactions was that it's not necessarily the case. You could have cells which are stably maintaining a hybrid state, which can couple the ability to migrate as well as adhere, therefore allowing for a collective cell migration or clustered cell migration rather than individual cell migration as the only option. So we have been looking at, we and also other colleagues in the field have been looking at what is the association of these hybrid states with resistance to different drugs. Um, at least what we have observed is that these hybrid states can also be resistant to tamoxifen. Again, going back to the previous example that I gave you. Um, Another colleagues in the field have been looking at and have observed that these hybrid cells can also be resistant to different kinds of immunotherapy. So they have done these kinds of experiments where um, cancer cells in different phenotypes, epithelial, mesenchymal, and hybrid, were co-cultured with T cells and or with immune cells. I'm not sure it was T cells, but with immune cells, and they observed how many cancer cells were killed. And again, the hybrid cells were not as effectively killed as the other phenotypes. So there are these kinds of experiments, and in individual systems, some molecular targets have been identified based on which you can alter the behavior of hybrid cells, but it is still an open question how to target these hybrid cells in the clinic uh, without impacting the normal functions as much as possible.
Going back to the non-genetic mechanisms, you explained that there are some non-genetic mechanisms that can lead to this development of resistance. Could you also identify what were the driving factors that led to this push from a state to the other? Were you able to identify some of these in your studies?
Are you talking about the hybrid states cells, or broadly speaking, the non-genetic mechanisms?
Yeah, broadly speaking, so were there some molecular signatures that were responsible for the switching from epithelial to hybrid state or hybrid to mesenchymal or vice versa?
Sure. So at least the interactions between, or the conversions between epithelial, hybrid, and mesenchymal, they can happen due to multiple factors, such as hypoxia, for instance. Lack of oxygen can usually has been shown to trigger epithelial to mesenchymal transition, either a partial or a complete transition. Also, change in the mechanical environment around the cell, so stiffness of the matrix, the extracellular matrix, can also trigger cells to go from epithelial to mesenchymal. What can trigger the cells from mesenchymal to epithelial is much less studied and therefore much less understood as well, because that's usually the last step of metastatic cascade, when cells have already reached a distant organ and now they need to convert back to settle down there to colonize that organ and to regain their cell adhesion, and they compromise the ability to migrate and invade, because that's not really needed at that point of time from the point of view of cancer. So all these different microenvironmental changes. Also, there have been studies that sometimes the therapies that are being used in the clinic, they can also have a side effect, and they can induce EMT, or epithelial to mesenchymal transition. This has been observed in radiotherapy. This has been observed in targeted therapy, including tamoxifen, and other drugs that we are currently investigating in more detail.
So, if you were a clinician and you had to decide that what a patient should be given, which drug a patient should be given, can you explain your thought process? How would you go about it? Starting from a person's, let's say you have the genomic profile of a person, you have the sequence of the person. How would you understand what drug a person or what combination of drug a person should be given so that there is minimum relapse or minimum chances of drug resistance?
It's a very difficult question, and I'm sure clinicians face this on a daily basis. And there's of course a strong human value component to this question as well, and ethical component to this question as well. Um, so it's basically a mix and match kind of, from whatever I've understood speaking to clinicians, that let's say you can give them as much information as you can, be it the genomic profile, be it the previous treatment that the patient has already undergone, be it the sort of current readouts of the clinical readouts of the tumor itself, the stage, the grade, and so on. And in that scaffold, one also sort of looks at how much the disease has already progressed and what are the different options available. So let's say even if you have the genomic profile, are those genomic variants targetable? Do we have a drug against them? And if yes, then is that necessarily going to benefit this particular patient? Because there are the sort of genomic landscape is not driven by one mutation. There are multiple mutations, and there have been instances of this, that say mutation in gene A, based on which, let's say, if you decide to give drug X, but that might be offsetting the mutations in gene B, right? So in that case, whatever drug decision was taken might actually be not as effective, right? So it's a mix and match scenario. And the evolution of drug resistance, I think, is a fundamental process, which is basically sort of, if I were to speak colloquially, built on our intent to live, right? Or if you break it down at this level of a single cell, it's trying to survive no matter what the stress that you give. And so the drug resistance would eventually come in one form or another, most likely. What we are trying to do is, can we minimize that chance, and can we delay that chance as to the extent possible, that it does not blow up into a full-blown tumor yet again. Again, some studies that we and other colleagues have done have shown that the current therapies itself can aggravate tumor outcomes in certain scenarios, because you're applying very strong survival stress on a subset of cancer cells, and they may accelerate whatever paths that they might not have necessarily unlocked at that point of time, to unlock those. And some of those paths actually can lead to worse outcomes, such as metastasis. So it's a very complex process, the dynamics of which is much less understood even at a fundamental preclinical research level. And to take research from a preclinical level to clinical stage, when you have a patient sitting in front of you, is anyway a very, very difficult process. So the overall idea that, can we bring drug resistance to zero? I don't think that's the answer. The answer, or the framework in which we should be looking at this, is can we identify different exit routes that a cell is capable of taking, and then see how can we prevent that from taking for as long as possible, without necessarily trying to apply a very strong sort of maximum tolerated dose kind of paradigm, because that has other side effects on the quality of life of a patient as well, which often is not considered in when you report multiple clinical statistics, right? You say that the 5-year survival rate increased. Okay, sure, but what about other factors? It's not just about living; it's also about how a given individual is able to live. Whether those extra life that was added was really of the quality that patient would have preferred to, or not.
Very interesting. So from what you are saying, I'm able to understand that we are monitoring only one axis. We are only looking at one axis of the patient's life, but we are not looking at the other effects that it can have, the drug can have on its patient's health span, not just the lifespan, right? So that is something that maybe in future we could look at. In fact, there are people talking about personalized medicine, like designing the combinations depending on each person's profile, epigenetic profile, genetic profile, and so on. So how far do you think we are in that direction, and how far we still have to go? What are your views on that?
Right. So again, I mean, I see it as a mix and match problem with more options available, right? You have earlier there was only genetic profile available. Now there is additional epigenetic profile available, and the hope is that with all these layers of data that is being collected, it would be converted to useful, actionable information. And that is a fundamentally difficult question to answer, because we do not have causality at this stage. We have associations, right? We have we have probabilities that, given this variant is present in a given set of patients, then what are the different possible outcomes? And then, of course, for whatever variant, if there is a targeted therapy available, then one proceeds with giving that particular aspect. So certainly, it is this paradigm of personalized medicine, appreciates the complexity, and appreciates that cancer is not one disease. Cancer is a set of diseases which may manifest very differently in different patients. Um, but I think it still lacks, largely speaking, uh, the framework of thinking about it as a dynamic process and thinking about it from the perspective of what a living system can go through in terms of trying to evolve strategies to survive. And that appreciation, I think, needs to be brought in with much more, say, thought processes, frameworks, colleagues from ecology, evolutionary biology, complex systems, physics, engineering, um, to come in and look at some of this data to suggest us potentially new experiments that can be done that can elucidate the different trajectories that a given complex system, both at the what's going on within a cell as well as at a population level in a specific spatial structure, what can they achieve as a consequence of underlying interactions.
So I was going through your webpage, and one thing which struck to me was you mentioned that your aim is to design therapeutic strategies using network level and dynamic understanding of molecular mechanism. I think it is very interesting that we are not just looking at a snapshot of the cellular state, we are looking at the dynamics of how the interactions evolve. And for that, you're using network level data. So could you please also explain a bit about how you go on with the network level analysis, and what are the different aspects that a network can inform us about a system, because your lab is mostly focused on a system and the disease that is cancer. So the perspective, that the approach that you're taking is systems approach. So could you also explain or emphasize the importance of systems level understanding of complex disease like cancer?
So most of the sort of previous analysis in the field has been focused on, let's narrow down to one molecule or a set of molecules which are playing crucial roles in enabling cellular function, particularly cancer cell function, but not as much normal cell function. And then let's try to use that as a knob to turn to alter the cellular behavior. Now, there are 20,000, 30,000 molecules in a cell, or many more than the types of molecules in a cell, perhaps many more than that. And they're interacting with each other in different complex manners, which can also be at different length scales as well as different time scales. So when we are going to pull one string out of it, so certainly that string was doing an important role, and that's why we observed some potentially beneficial outcomes in terms of changing the cellular behavior in this context of cancer. But what is not appreciated is that when you pull that string, the expected outcome is that a cell dies, a cancer cell dies, right? But there are other outcomes also that can happen because that same molecule whose string you pulled was actually impacting the behavior of many other molecules which were responsible for a different function. So again, in the example of tamoxifen, we have seen that the tamoxifen treatment can certainly kill a subpopulation of breast cancer cells. But in the other outcome that is possible as a consequence of repression of estrogen receptor signaling pathway, which is what tamoxifen targets, is that it could make cells more metastatic. Now, what we do not know yet is that what is the percentage of cells that are being killed versus becoming more metastatic? How does this depend on, say, the dose or the duration of tamoxifen given, or other previous treatments given, or the genetic makeup of a patient, and so on and so forth? So those are variables we are still trying to investigate how they impact these. But the central underlying philosophy in the way we are approaching the question is that we should be looking at different possible outcomes of a given perturbation to a system. One, of course, is cell death, which is what everyone has been focusing on. But let's look at the cells that do not die. What prevents them from death? And can they become more aggressive as a consequence of the stress being applied? And in certain examples, including tamoxifen, we have seen that yes, that can indeed be the case. So once we know the different possible outcomes which can emerge as a consequence of interactions among different molecules in a cell, then one can think of making a more informed choice in terms of which combinations of drugs may actually cancel out each other's potential side effects, in terms of the other possible outcomes at a cellular level, and perhaps be a better strategy as compared to both of those drugs alone.
Very insightful to understand how the systems approach is helping us to solve a real world problem. So I think systems are everywhere. If you look in economics, in politics, in biology, also if you look at large scale migration of animals and so on. So one final question I would like to end with is, why did you choose cancer as a system? Was there something personal to you, or was it challenging to you that you found that maybe I should tackle this as a challenge? So what brought you to the cancer systems particularly?
So I was interested in, broadly speaking, systems biology. Just I identified that is what I wanted to do, at least for my PhD, and I happened to read this book, Cancer, the Emperor of All Maladies by Dr. Siddharth Mukherjee, which is a wonderful book. I enjoyed reading it and realized that we know so little about this disease, despite its existence for, at this point, centuries altogether. So that really motivated me that, is there a possibility that by using systems level approaches, can we figure out some fundamental aspects about how cancer proceeds, about how cells change their behavior to undergo metastasis or to undergo to develop resistance to different drugs. And so that's what I would say was my major motivation. And then, of course, while I started my PhD and was thinking about some research questions to address, I would see many cancer patients at MD Anderson Cancer Center right across the street from Rice University in Houston on almost a daily basis when I would go there for seminars or other collaborative meetings and so on. And that angle was always there at the back of my mind. Is there something we can do to improve the quality of life for this huge population of cancer patients? And again, as you would have noticed, the percentage in India is also increasing in terms of the incidence and deaths caused by different kinds of cancer. And we are still not there as a community, as a scientific community, to be able to cater to all the different challenges that are being faced in the clinic. So that's the motivation that keeps coming. It was triggered by that book, but then many real life stories of people I have known, and the second and third degree connections as well, really continues to motivate me to work on these challenging questions in the clinic.
That's so awesome to hear. A book can pave a path for a person's life and trajectory. We hope that this talk also motivates some of the people to work on some of these challenging problems, not necessarily cancer. Maybe they could identify something else. Like you said, you read a book and you also had some context, the real world context, to understand what are the challenges. Maybe the people who are listening to us to this podcast, they would also get inspired by your journey. How far you have come. You have won so many awards that if I go on listing, half of the podcast would be spent in that. So congratulations on winning so many prizes, and we hope that you continue making the impact more than the prizes. The impact that you're making is more important. And thank you for giving your valuable time. Thank you very much, Professor.
Thank you so much, Anjali. It was lovely talking, and looking forward to chatting again soon. Thank you.
Thank you.